Thank you very much for inviting me today. I'm really happy to talk about this very recent preprint. Actually, if you scan the QR code, this will lead you directly to the manuscript, and it's a perfect follow-up for the talk that we just heard. Actually, it follows the same line, I would say. And normally when I give the presentations, I always have to go very deep on the introduction, but here luckily it's not needed regarding TCR repertoire. The only thing that I would like to say is that we don't have enough, I think, public data set, including alpha chain and not only beta chain, especially for the bulk sequencing. So this big data set that there are outside, like the Emerson data set, for example, they only have the beta chain, and this starts to be a bit limiting for certain applications. But today I will tell you a lot also of data done with single cell sequencing, which of course kind of solve the problem, at least partially. So what we did briefly in this small work, which was done in collaboration with Michael Pogorelli and Anastasia Minervina, which are now at the St. Jude Hospital in Memphis. We took in practice two big single cell data sets that contain only CD4 T cells, all reactive to SARS-CoV-2. These two data sets are nice because they were produced with the same method, but at two different locations. So one is our own data set produced in Kiel, and the other one is a data set from McKeith and colleague. And we merged the two data sets, and we obtained this big data set containing all single cells reactive for SARS-CoV-2. Then independently, we took two big TCR bulk data sets. So the Emerson et al. With the only healthy controls, and Snyder et al., which contains SARS-CoV-2 patients. And then in practice, we identified statistically using these two big bulk data sets, TCRs, which are enriched or depleted actually in COVID-19 patients. And then we cross-referenced the two things. So we cross-referenced the single cell data set with the two big bulk data sets, and we kind of went to characterize as deeply as we could the TCRs. So what we wanted to do is to not only find the TCR alpha beta pairing, but do a bit more deep characterization. So find also the HLA type, the HLA specificity, the precise antigen specificity, and as a plus, perhaps the phenotype. And this was a bit our plan. And why is because there is one main method with which we identify T-cell epitopes nowadays, and this is what we call the standard somehow epitope discovery, which is you have your antigen of interest and you stimulate T cells, so you find out, okay, these T cells react to my antigen. But now that we work with these big TCR data sets, there is more and more this will somehow of having the contrary. So starting from a list of TCRs and say, okay, this TCR is specific for this TCR is specific for that. And this is somehow what we call reverse epitope discovery, which is super interesting and also super difficult somehow. And one thing that we thought is, okay, this is a very nice method. It doesn't go to substitute the standard epitope discovery, of course, but it's a wonderful complementary method if we can make it work. So what we want to have in the end, our dream, I would say, in our field is to have a list of TCRs and be able to give the specificity of each of them. What we did here is to study, again, this definition of public clonotypes, so clonotypes that are present in many people, which of course are somehow a very good and a bad thing, because on one side, they give you a very good statistical power because they are public. On the other side, by doing this, we somehow miss all the signals that come from private TCRs. And of course, because they are public, we thought they can also have somehow a potential for broader applications because they are found in so many people. And again, what we did is this deep characterization, somehow the HLA antigen paired alpha beta TCR axis. And the idea is that while doing this characterization, you can, of course, then study much better certain specific T cells, because if you know the HLA and the antigen specificity, you can also capture, again, the specific T cells or re-express these TCRs and do more experimental validations or experimental analysis. And as a bit futuristic things, perhaps, but not even too much, the characterization very deeply of TCRs, I think, may be a useful topic for adoptive T cell transfer, and usage of specific TCRs into clinical practice somehow. So now I go to explain in detail a bit what we did. So as I said, we have these two big single cell data sets that merge together. We have around 125,000 cells for which we have TCR for all of them. And as a basic thing, we want to first re-identify all the phenotypes of these cells, which you see here. So this is the typical COVID-19 response phenotype in CD4 cells. So we have the follicular T cells, the Th1, Th17, effector memory, central memory, interferon signal, and the cytotoxic signal. And then, as I said, we took these two big data sets of bulk TCRs, and doing simply a Fisher test, we identified which ones are enriched, statistically enriched, and statistically depleted in COVID-19 patients. And you see them here. The purple ones are the ones that are statistically enriched, and the green ones are the ones that we found to be depleted. And you see it here, the fraction of donors in which we find these clonotypes, and you see the difference between somehow COVID patients and controls. Then among these COVID enriched and depleted, we retained the ones that are found in the single cell dataset. So for these ones, we now have the alpha and the beta chain. So it's interesting to notice that we just retained the somehow public ones. So most of these COVID enriched that we retained are found in at least 100 individuals. And when we went to cross-reference with the single cell dataset, we found multiple people, even in the single cell dataset that contains just 50 people all in all, that had the same beta and in some cases even the same alpha chain, or a sequence that was differing by just one amino acid, for example. And then with this list of clonotype, one thing that we wanted to see is, do we find somehow a specific or a preferential phenotype of these clonotypes? And the difference that we see is that in the clonotypes that are enriched in COVID-19 patients, these are also enriched in the follicular T cell compartment, while the clonotypes that we found depleted in COVID-19 patients are somehow enriched in the effector and central memory phenotypes. So the reason why we find somehow COVID enriched clonotypes, and these are in the follicular compartment, is somehow we think these are the ones that are mostly responded to the disease, or that drive the response to the disease. And we have an explanation why we also find these COVID depleted ones that are a bit somehow more puzzling. And I don't go into details now, but I'm happy to take questions later if you're interested. But for the next analysis, we focused on the ones that were enriched in COVID patients. And what we wanted to do is to, as I said, characterize them, find the specific antigen and HLA type. So what we did goes along the lines of the TCR clustering that was described in the previous talk. And we clustered the TCRs based on their amino acid sequence. The interesting thing is that we did a clustering that is not based only on alpha or beta, but uses both chains, and with a software that is called TCR-dist. And this is the clustering representation. So each groups connected together represent a cluster of TCRs that differ by somehow, let's say, 2 amino acids, but they are assigned a so-called TCR distance, exactly. And one can play with the threshold of this distance and somehow find groups of TCRs that have very similar alpha and or very similar beta chain. Then using the data set that was also described in the previous talk, this MIRA dataset made by Adaptive Biotechnology, we were able to identify the specificity of these TCRs. So this dataset doesn't give the specific peptide to which the TCRs are reactive, but gives collection of peptides from a specific protein. So it's like, I don't know, 5 to 15 peptides per peptide pool. And this is what you see with the colors here. So for example, the green ones are TCRs that are known to be reactive to a peptide pool of the membrane protein of SARS-CoV-2. The yellow ones are the pool of the spike protein, and so on and so forth. So you see the colors and the assignation at the left here. And then we went to characterize in deep some of the biggest of these clones that form this very big TCR response. And we did first the HLA specificity assignation, and this also we did it statistically. So luckily the HLA type of the individuals of the Emerson collection is known. So thanks to this, we identified the HLA specificity of most or many of these TCRs, and also for part of the single cell data set we have the HLA types, so we actually checked that this was corresponding. And then we used NetMHC to identify the strongest binders, peptide binders to the identified HLAs among these peptide pools that were known to bind to these TCRs, and that's what you see here. So here, I know the figure is a bit complicated, but each row it's one of the clusters. So for example, if you look number 2, it gives you the alpha chain motifs. So this is the B gene of the alpha chain, the J gene of the alpha chain, the CDR3, and the same for the beta chain. And at the right, it gives you the predicted HLA specificity and the pool to which some of these TCRs are known to be reactive, and the sequence of the specific peptides that we predict to be the real binder. And this we did for these 6 big clusters. A few, I would say, interesting points is that, for example, not all of these clusters are driven by somehow only beta or only alpha similarity. You see that cluster 1 has a quite defined motif that uses a fixed B alpha gene and also the same B beta genes, but there are other clusters that are not so obvious. So the cluster 2 is strongly driven by the alpha chain similarity, for example. And other clusters are driven only by the beta chain, like cluster 3. It's very diverse in the alpha, but very similar in the beta, while the other clusters are more driven by both chains. This is something that personally I found quite interesting. And then one point is also, okay, how can we validate this prediction that we did? So we predicted the HLA specificity, we predicted the antigen and peptide specificity, but how do we know if this is correct? And especially what we wanted to do here is, okay, for the green ones or the colored dots, so to say, we know that at least they should be reactive to a certain peptide pool, but the gray ones don't have any annotation in literature. So one of the things that we find positive of this method is, okay, then you can try to annotate the specificity of what we call orphan TCR. So, and with orphan, we mean TCR that don't have any specificity annotation in literature. Luckily, while we were working on this thing, the two studies came out that independently validated some of our predictions. Let's see. Yeah, and as you see here, this cluster 2 highlighted in red, it was recently accepted in Cell, this paper by Matt et al. And what they did, they analyzed the axillary lymph nodes following SARS-CoV-2 vaccination. And they found that the follicular T-cell response is strongly driven by a big TCR cluster. And by coincidence, this big TCR cluster is exactly this one that we find here. So they experimentally validated the specificity, and it is exactly what we predicted here. And the same thing we found in Lu et al., who studied a certain number of TCRs and analyzed the specificity. And among those TCRs, there was one or two of these cluster, which is also specific for the spike protein. As a last thing, we took another data set that contains TCR, bulk TCR data pre and post vaccination with the AstraZeneca vaccine. And we checked if just by using the clonotypes included in these clusters, we could see a difference between pre and post vaccination. And that's what we saw here, that a statistical difference in the presence of clonotypes is seen only for cluster 2 and cluster 5. And cluster 2 and cluster 5 are the only ones that are predicted to bind the spike protein, which is what we expected and we wanted to see. So all in all, we identified these highly public CD4 T-cell responses to SARS-CoV-2. And we wanted to see how, or to show how this reverse epitope discovery method can be used and how to identify the specificity of, here I write public clonotypes, because as I said, we analyzed public clonotypes, but the idea is that we want to find the specificity of orphan TCRs. And for sure, there are a lot of private orphan TCRs. And once we assessed somehow that the computational method works and is trustworthy, the idea is that one can analyze or find the specificity of only one or two of the TCRs in certain clusters and automatically somehow assign the same specificity to the other TCRs in the cluster. Just for safety, we are doing also our own experimental validations of these predictions, and spoiler alert, it seems to work pretty nicely, luckily. And as a last thing, of course, we hope somehow that this prediction of specific epitopes can also be of some use of whoever wants to use the data. It will all be published, and including all the scripts will be deposited on GitHub for the method we use. And with this, I want to thank all the people involved in the project. So first of all, as I said, it was really a 6 hands work with Misha and Dasia in Memphis. And all my group in the Institute of Immunology and the Institute of Clinical Molecular Biology in Kiel. You see this as my group. And of course, yeah, all of you for your attention. I'm looking forward for questions.
Thanks so much for this beautiful talk. Are there questions from the audience?
Yeah, I have a question. So how do you interpret cluster 1? Because it seems to have 3 different specificities across the genome. And so do you think this is real cross-reactivity or something else? What's your...
This is one thing that it's a very good question, and it's I mean, it's normal that it comes up because it looks very weird, right? So these are all TCRs that in a Myra dataset are assigned to, so the green one to the membrane, the yellow one to the spike, and the purple one to the nucleocapsid. And I also was very puzzled by this. I thought it may be cross-reactivity because there are other TCRs in the Myra dataset, which is the same TCRs assigned to multiple pools of different proteins. So that may be cross-reactivity. Another thing that I thought is there is only the beta chain. So maybe they have different alpha chain, they have different specificities. All those, I don't know how this is likely. And the third option, which I think is the most probable one, it's somehow a technical error, because the way that the MIRA assignation is done, it's somehow also is of disentangling the specificity so the stimulation is done with different pools also all together, and then they disentangle the specificity of them. And it may be that there is then a misassignation of the specificity. And we thought that this is the most likely thing, I would say.
If I can comment on that, I would not think so. I think it's, we call this polyspecificity. And we've studied, for example, T cells from COVID-19 patients isolated from bronchoalveolar lavages. We found some T cells with TCRs that had been assigned to SARS-CoV-2 before, but also TCR that had been assigned to response to CMV, EBV, and others. And we have a bioarchive paper that we have a hard time to publish, in fact, which is describing this in more detail. And I'll be happy to discuss this with you. It would be a long discussion, so maybe not here. But we think that this represents a relevant biological phenomenon that, you know, we think is worth investigating in more details.
Yeah, so that was somehow, yeah, really fascinating, because I don't know how likely it is for the same TCR to recognize 3 different proteins, or like different proteins in the same virus. In this case, it's different TCRs which are assigned to different proteins. So it may be that one of them really recognizes a nucleocapsid, and the other one, although very, very similar, recognize a different protein.
Well, so we have actually identified and cloned these kind of receptors and expressed them in reporter cell line, and showed that they respond to very different unrelated peptides. And in fact, Mark Davis published recently in Immunity his paper where he looked at the TCR response, the T-cell response against lung cancer cells, and he identified like this a dominant TCR that responds to a self-tumor antigen that he actually identified, but also responds to an E. coli peptide and an EBV peptide. And he also cloned these TCRs, expressed them in reporter cells, and showed that they even respond at a smaller concentration of the peptides against coli and EBV than it responds to the self-antigen. And we're seeing exactly the same thing. I think that there's something that people don't want to look at, which is that overlooked under the specific response, which of course do exist. There are TCRs which are actually responding to multiple unrelated, peptides, and I think it's worth investigating. I may be wrong, but I think it's worth investigating.
Yeah, of course. I mean, that's why I like to say somehow that TCRs are antigen reactive and not antigen specific, because we know cross-reactivity is a very important point. And of course, it's also fairly easy to assess this cross-reactivity in this case, in sense we are re-expressing the TCRs in a Jurkat cell line, and one could then see if they really, as you also said, react to multiple things.
Yeah.
Any more? Yeah, so we have one question also in the chat. Elisa, so in the chat, Alexandra asks, what maximum number of substitutions indels do you allow to assign the specificity of TCRs in a cluster?
Yeah, so as I said, it's a very good question, of course, very important. And this is done somehow internally by the software. So this TCR dist software. Normally, when I do it, manually, I do it maximum like only using the Hamming distance. So we just take TCRs that have the same length of the CDR3, and I take then maximum 2 mismatches in the CDR3 region. In this case, it's done by the software. So I don't know the exact parameters that we used, but is the software who somehow assigns the similarity of the of the TCRs. And I know that we were actually pretty conservative here with the distance. So you get a score at the end, a distance score between TCRs. In this case, we used, it doesn't say anything, but we used 120 as a score. And we were pretty conservative also because somehow of the errors in the next genomics single cell things, where you can have doublets and stuff, and there were somehow some clusters where you were reducing the distance, or actually augmenting the distance, that were clustered together, that were obviously not supposed to be. So we reduced the distance to be as conservative as possible. So yeah, I cannot answer the question exactly, but I suggest the TCR, this paper, this should be more clear.
Great. Then one question from me. So you mentioned these depleted clonotypes also in the beginning, but you didn't go further into detail. Can you explain a bit more what you think about them?
Yeah, we were we were really puzzled by this, because we were like, okay, depletion, why should be specific TCR selectively depleted, right? And then we checked, actually we came out of the idea of lymphopenia as a reason for this, because actually we checked, and most of the COVID patients have less clonotypes than the healthy controls. And this seems to somehow bring to some statistical difference in the presence of very, very public clonotypes. So these COVID depleted ones are all clonotypes that are present in more than 100 COVID patients and more than 100 healthy controls. So they are really public. The effect of the difference between COVID patients and healthy controls is not as strong as in the COVID enriched. So in the COVID enriched, the statistic, the p-values were much lower, while in the COVID depleted, it was not a strong statistical difference. And that's the explanation that at least we gave to it, that normally you would not see a difference in the presence of these ones, but probably because of lymphopenia, you do see a statistical difference. But I would say that's a plausible explanation, but it was also a bit, puzzling.
Do you know of other infections or diseases where this has been observed, such immune status specific depletion, or is this for COVID the first time observed?
Yeah, I also was thinking about that, to be honest, and I couldn't come up with an answer. Probably I didn't look enough into the literature, because I would think that this is something very normal, but it's not only, I would say, disease specific, is also like I mean, somehow data driven, because if you have less clonotype, if you capture less clonotypes, then I would say you risk to miss something that you would normally see.
Yeah. So it's a bit observation bias or study bias also. So we need to see more also. Yeah.
I would say so.
Yeah. Great. Any last questions for Elisa? David, you are muted. You have to unmute.
So there is a question in the chat.
Yeah. But first David, and then the
David.
Yeah. David?
Oh, yeah, no, I just commented also that depletion in peripheral blood may mean actually that the cells are elsewhere and actually concentrated in tissues. So it's
that's one option. On the other side, I must say that these COVID depleted ones, so we tried also, and they were clustering much less based on TCR similarity. And also the interesting thing is that they were somehow enriched. So also in the single cell data, they were somehow more present in healthy controls of the single cell data, also compared to the COVID-19 patients, although in the single cell data, we have much more cells for the COVID-19 patients compared to the healthy controls. And they also, the fact that they also are in different functional clusters somehow, or preferentially, it's also quite interesting, I would say.
Okay, last question and very short answer, please. Did you also investigate CD8 cells?
Yeah, so yes and no, in sense that it would be nice, but so the one point with this data is that we need a lot of data, and we need the bulk, we need the single cell, we need the antigen pool specificity. This was possible now because of the big amount of data that were produced because of the COVID pandemic. But for the CD8, there is only one data set of single cell CD8 cells. And somehow we couldn't be sure of the specificity because the stimulation time, in our opinion, was done a bit too long to be sure of the specificity. So we decided to don't work with this data and to do it only CD4 for this study. But I would assume normally it should of course work also for CD8.
Great, and that's a nice final word. Thanks again so much to Thierry and Elisa for their very nice talks. We will come back to the second installment of this year's seminar on February 24th, so in 1 month. Thanks again so much, everyone, and stay safe. Bye-bye, everyone. Bye-bye.
Thank you. Bye.
Thank you. Bye.